Our prototype, using cassava pulp to generate biofuel.
While aerobic respiration produces more energy per glucose molecule in the form of ATP production, we cannot access or harness this energy readily. We thus rely on anaerobic respiration of other organisms to harness the ethanol they produce as biofuel.
Cassava is a starchy crop that grows in tropical countries. It is commonly made into starch, with cassava pulp being a solid byproduct of the processing. Cassava pulp is a promising and underused biomass for ethanol fermentation, as it contains approximately 60% starch and 20% cellulose fiber. By adding enzymes such as pectinase, cellulase, hemicellulase, and glucoamylase that break down the starch and cellulose fibers through saccharification, we can obtain the simple sugars ready for fermentation.
A chute will open and the slurry of sugars will flow into another container, where yeast will be added. Here, through anaerobic respiration, where oxygen is not required, the yeast will ferment the sugars, turning them into ethanol and carbon dioxide. Under these anaerobic conditions, no further oxidation of pyruvate occurs, no acetyl-CoA is formed, and no additional ATP can be generated, which is why, as compared to aerobic respiration, in which oxygen is used, significantly lesser energy is produced. The content in the container is cooled down to about 30 degrees, the optimum temperature for yeast fermentation. The yeast breaks down the glucose into pyruvate acid. The pyruvate acid is then broken down into carbon dioxide and ethanol through two enzymes, pyruvate decarboxylase and alcohol dehydrogenase, within yeast cells.
Alcohol content that is produced stays in the container. Yeast cells can tolerate only a certain concentration of alcohol, and any more than their tolerance level would cause the yeast cells to die. After the carbon dioxide has been removed, the ethanol is distilled from the container. The vaporous ethanol will condense, allowing it to be collected for usage as biofuel.
Created using Powtoon.